Namaste everyone, I am your co-host Shravani ma'am. Welcome to your favorite learning channel. The topic for today's video is movement of substances by active and bulk transport. So in our previous video, we had discussed about the movement of substances that was taking place by the help of passive transport. In today's video, in continuation with that, we would be discussing about active transport and bulk transport which help in the movement of substances across the plasma membrane. Okay. So first we would begin with active transport. Okay. Active transport. So let's see what is active transport. So this active transport, it is actually a mode of transport. So it is a mode of transport that takes place across the cell membrane or across the plasma membrane. Okay. And this process of transport involves the expenditure of metabolic energy. So unlike passive transport where it required no metabolic energy for transport of substances, here the transport of substances involve or require metabolic energy, and this energy is obtained from where? It is obtained from ATP, that is adenosine triphosphate, provides energy for this process of transport that requires energy. Okay. So ATP provides the energy. So it is the most common method of salt absorption in plant cells. Okay, so active transport is the most common method for absorption of salt in plant cells. And this process occurs against the concentration gradient. So it occurs against what? Against the concentration gradient. So passive transport was occurring along the concentration gradient, whereas active transport occurs against the concentration gradient. And so it is also known as uphill transport, because it is taking place against the concentration gradient. Okay. So the cells that are engaged in active transport, they show a higher rate of respiration. So the cells would be able to accumulate substances against the concentration gradient. So it is unidirectional. This process is what? It is unidirectional. Okay, it is unidirectional, and also it is highly selective, and it shows a saturation effect. It shows what? It shows a saturation effect. Okay. So when the concentration of transportable solute is increased, what happens? The rate of transportation also increases, but as the concentration of the solute decreases, the rate of transport also comes down. Okay. So the decrease in oxygen availability decreases the rate of active transport. So rate of active transport decreases. It decreases when it decreases with the decrease in the oxygen availability. Okay, and the metabolic inhibitors like cyanide and azide stop active transport. Okay, and the decrease in temperature reduces the rate of active transport as well. So rate of active transport usually decreases with decrease in oxygen availability, and the decrease in temperature also reduces the decrease in temperature. Decrease in temperature also reduces the rate of active transport. And apart from that, there are metabolic inhibitors. So the metabolic inhibitors like cyanide and azide, what do? They stop active transport. They stop active transport. Okay, so this was about active transport. Now let's discuss about the bulk transport. Okay, so the next is bulk transport. Okay, so what is bulk transport? It is also a mode of transport. It is also a mode of transport, and this mode of transport is of large quantities of what? Of materials and food particles. Okay, and this happens across the membrane, that is across the cell membrane or plasma membrane. So it involves bulk transport involves what? It involves chemical stimulation. It also involves folding. Okay, there is folding, and also there is fusion of membrane. There is folding and fusion of membrane so as to produce what? So as to produce vesicles. To produce vesicles called the carrier vesicles. Okay, so this bulk transport occurs both inward and outward. So the living organisms take their food in bulk in the form of large sized molecules which cannot cross the plasma membrane. So these molecules are transported by two processes. So the molecules are transported by two processes. One is called exocytosis, and the other process is called endocytosis. Okay, so now let's see what is exocytosis. Okay, exocytosis. So exocytosis is also called as cell vomiting. Okay, so it is a process of expulsion Here what happens? In this process, there is expulsion. Expulsion of what? Expulsion of materials from the cell, and this happens with the help of carrier vesicle. With the help of carrier vesicle called the exocytotic vesicle. That is called the exocytotic vesicle. Okay. So it is common in secretory and excretory cells. Can be commonly occurs where? In the secretory or the excretory cells. So these secretory or excretory products are actually packed in vesicles by Golgi apparatus and are then passed outward. So later what happens? It comes in contact with the cell membrane. So the vesicle then fuses with the cell membrane and bursts open to release the contents of the cell. So exocytosis is important in throwing out waste materials. Why is it important? The importance is that, or we can say it is important because it helps to throw out waste materials. Okay, so it helps to throw out the waste materials out of the cell, and in the secretion of cellular products such as, and it also helps in secretion of cellular products. And what are these products? The products like digestive enzymes, or hormones. Okay, so for these purposes, this process of exocytosis is important. Okay, so now let's talk about the next process, that is endocytosis. Okay, so what is endocytosis? It is also a process. What happens in this process? In this process, the cells absorb materials. Such as proteins, from outside. They absorb them from outside by engulfing them. Okay, so it is used by all the cells of the body because most of the substances are large polar molecules which cannot pass through the plasma membrane. So endocytosis is performed by the cells, and this endocytosis is further classified into 3 types based on the nature of absorption of materials. Okay, so we can say that endocytosis is of 3 types. One is phagocytosis. The next one is pinocytosis, and the third one is called the mediated receptor mediated endocytosis. Okay, so these are the three types of endocytosis, and this classification is made based on the nature of absorption of the materials. Okay, so first let's talk about phagocytosis. Okay, so this word phagocytosis means cell eating. Okay, and this cell eating is the meaning of the word phagocytosis, in which the cell engulfs. Here what happens? Cell engulfs. What does it engulf? It engulfs bacteria, debris, or any other sizable objects. Okay, so it is the process of absorption and ingestion. It is also a process, and it is a process of absorption and ingestion of particles. Okay, it is a process of absorption and ingestion of particles by the cell and by many protists, such as, so by the cell as well as by many protists such as like amoeba. Okay, so amoeba performs phagocytosis to acquire nutrients. So this process of phagocytosis was discovered by a scientist. It was discovered by a scientist named Mechnikov. Okay, so the scientist was Mechnikov, who discovered this process of phagocytosis, and we can define this process as, how can we define? So we can say definition of phagocytosis. How can we define? It is a process of ingestion of large particles by engulfing. This is how we can define the process of phagocytosis. So this phagocytosis occurs in specialized cells. So phagocytosis occurs in specialized cells known as phagocytes. Okay, and these phagocytes could be, for example, neutrophils, then macrophages, and various other white blood cells. Okay, so the cell membrane of phagocyte actually possesses receptors. So the particles attach to these receptors The microbes bind to receptors through antibodies produced by the body. The edges of the cell membrane may invaginate or surround the particle, rapidly making more contacts between the receptor and the surface ligands of the particle, and this produces a vesicle which is then pinched off with the help of actin microfibrils, and the vesicles containing the engulfed solid particle are called phagosome. Okay, so phagosome is the phagosomes are the vesicles that contain engulfed solid particle. Okay, there is another term, phagosome. They are the vesicles that contain engulfed solid particle. Alright, so these are called phagosomes. So one or more lysosomes fuse with phagosome to form food vacuole. The digested material then diffuse out of the vacuole, and the food vacuoles are left with indigestible materials, and these form a residual vacuole. The content of the residual vacuole are thrown out through the process of exocytosis. Okay, so this was all about phagocytosis. Now let's talk about the next process, that is pinocytosis. So pinocytosis means cell drinking. Phagocytosis meant cell eating, whereas pinocytosis means cell drinking. And how can we define this? Definition. We can define pinocytosis as a process. It is a process by which the cells uptake solutes and they uptake single molecules. They uptake single molecules like sugars and proteins. So this process is known as pinocytosis. So this process was discovered by the scientist Edward Lewis. Okay, so Edward Lewis was the scientist who discovered the process of pinocytosis. It helps to intake solutes such as insulin and lipoproteins in concentrated form. So when the extracellular solutes attach to the specific receptors of the membrane, an invagination is formed. Now this invagination is drawn inwards and it is pinched off into small vesicle called pinocytic vesicle or pinosome. The pinosome separates from plasma membrane and then passes into crystal matrix for either bursting or to fuse with tonoplast. And sometimes the pinocytic vesicle passes into cytoplasm where they fuse with lysosome, and it leads to the formation of digestive vesicles to reach Golgi and to deliver its contents. Okay, so this is the whole process of pinocytosis. Okay, and then the third process is the receptor mediated endocytosis. Okay, so what is receptor mediated endocytosis? It is also a process. In this process, specific molecules are ingested. Here the specific molecules are ingested. Okay, and they are ingested into the cell. So the specific molecules are ingested into the cell, and the speciality is based on the receptor ligand interaction. What is the speciality? The speciality is based on the receptor ligand interaction that we can find in the receptor mediated endocytosis. So the receptors that are present on the plasma membrane of the target tissue will specifically bind to ligands present on the outer side of the cell. An endocytotic process occurs and the ligand is then ingested. Okay, so this is the process of receptor mediated endocytosis. So now let's quickly revise everything that we have studied today. So today we discussed about the movement of substances that occurs by the help of active transport as well as by the help of bulk transport, and under bulk transport we studied about endocytosis and exocytosis. So first we discussed about active transport. Active transport is a mode of transport that occurs across the cell membrane, and it involves the expenditure of metabolic energy, and this energy is obtained from ATP. So ATP, that is adenosine triphosphate, provides the energy that is required for the process of active transport to take place. Okay, so this is the most common method of salt absorption that can be seen in plant cells, and it occurs against the concentration gradient and not along the concentration gradient as seen in the case of passive transport. And as it occurs against the concentration gradient, it can be called as an uphill transport. That is, active transport can be called as an uphill transport. So the cells engaged in active transport show a higher rate of respiration, and the cells would be able to accumulate substances against the concentration gradient. So this mode of transport is unidirectional and is highly selective, and it shows a saturation effect. So the rate of active transport decreases when there is decrease in oxygen availability, also when there is decrease in temperature. Then there are some metabolic inhibitors like cyanide and azide that can stop the process of active transport. Okay, so when the concentration of transportable solute is increased, the rate of transportation also increases, but as the concentration of the solute decreases, the rate of transport also comes down. Okay, so this was about the active transport mechanism. Then we discussed about bulk transport. So bulk transport is a mode of transport. Okay, and in this mode of transport, large quantities of materials and food particles are transported across the membrane. So what does it involve? It involves chemical stimulation, folding and fusion of membrane so as to produce the vesicles that are called the carrier vesicles. Okay, so bulk transport can occur both inward and outward, and the living organisms take their food in bulk in the form of large sized molecules which cannot cross the plasma membrane, and these molecules are then transported by two processes, that is exocytosis and endocytosis. Then we discussed about exocytosis. So exocytosis can also be called as cell vomiting. It is the process of expulsion of materials from the cell with the help of carrier vesicle called the exocytotic vesicles. Okay, so it is commonly seen or commonly found in secretory or excretory cells. Okay, then the secretory or excretory products are actually packed in vesicles by Golgi apparatus, and then they are passed outward. Later what happens, it comes in contact with the cell membrane. The vesicle fuses with the cell membrane and bursts open to release the content of the cell. So exocytosis is important for throwing out the waste materials out of the cell and also in the secretion of cellular products like the digestive enzymes or the hormones. So in these processes, exocytosis play an important role. Okay. So then we discussed about endocytosis. So endocytosis is a process, okay, whereby the cells absorb materials like the proteins from outside by engulfing. So this endocytosis is used by all the cells of the body because most of the substances are large polar molecules which cannot pass through the plasma membrane. So endocytosis can be further divided into three types, and this classification is based on the nature of the absorption of materials. So the three types of endocytosis are phagocytosis, pinocytosis and receptor mediated endocytosis. So we can now revise this with the help of a diagram. Okay, the three types of endocytosis. So first is phagocytosis. So phagocytosis means cell eating. So what happens here? Here the cell engulfs bacteria, debris or other sizable objects. So it is actually a process of absorption and ingestion of particles by the cell and by many protists like amoeba, by which the amoeba acquire its nutrients. So the process of phagocytosis was first discovered by the scientist Mechnikov, and we can define phagocytosis as the process of ingestion of large particles by engulfing. So phagocytosis occurs in specialized cells and these cells are called as phagocytes. Okay. So the phagocytes could be neutrophils, macrophages and any other white blood cells. So what happens? The cell membrane of phagocyte possesses receptors. Okay, there are various receptors present on the cell membrane of phagocyte, and the microbes bind to receptors through their antibodies produced by the body. Okay, so the antibodies that are produced by the body, with the help of that the microbes bind, and the edges of the cell membrane invaginate. Okay, so the cell membrane then invaginates, okay, or they surround the particle rapidly, making more contacts between the receptor and the surface ligand. Okay, it surrounds the particles rapidly. Here we can see this is the invagination. Okay, so this produces a vesicle which is pinched off with the help of actin microfibrils. Okay, a vesicle then forms that later gets pinched off with the help of actin microfibrils, and the vesicles containing the engulfed solid particle are called phagosome. This is the complete vesicle, okay, and it is containing the engulfed solid particle. First there was an invagination, then it got pinched off and it led to the formation of phagosome. Okay, so one or more lysosomes fuse with phagosome to form food vacuole. Okay, so the digested materials diffuse out of the vacuole and the food vacuoles are left with indigestible materials forming a residual vacuole, and the contents of the residual vacuole are thrown out through the process of exocytosis. Okay, so this is the process of phagocytosis. Next we will discuss about pinocytosis. So how can we define pinocytosis? So pinocytosis can be defined as the process by which the cells uptake solutes and single molecules such as sugars and proteins. And the word pinocytosis means cell drinking. So this process was discovered by Edward Lewis. It helps to intake solutes such as insulin and lipoproteins in concentrated form. So what happens when the extracellular solutes, this is extracellular fluid, and when the extracellular solutes attach to specific receptors of the membrane, an invagination is formed. Okay, and this invagination is drawn inwards or and gets pinched off. Okay, so this invagination later gets pinched off into small vesicle called pinocyte. Small vesicle develops, that is called pinocytic vesicle or pinosome. The pinosome separates from the plasma membrane and passes into crystal matrix for either bursting or to fuse with tonoplast. Sometimes what happens, the pinocytic vesicle passes into cytoplasm where they fuse with lysosome to form digestive vesicles, or they reach Golgi to deliver its contents. Okay, so this is the process of pinocytosis. Then we had also discussed about receptor mediated endocytosis. So it is the process in which specific molecules are ingested into the cell. So here only the specific molecules are ingested, and the speciality is based on the receptor ligand interaction, hence the name receptor mediated endocytosis. So the receptors on the plasma membrane of the target tissue will specifically bind to ligands. Okay, there are receptors which on the plasma membrane which specifically bind to the ligands present on the outer side of the cell, and an endocytotic process occurs and the ligand is then ingested. Okay, here we can see coated vesicle, and these are the coat proteins. Okay, so this is the process of receptor mediated endocytosis. So I hope you all would have got some great insights by watching today's video. Please do not forget to like and share the video. Till then, thank you for watching.